We, we're going to talk about enzymes today. Enzymes are protein molecules. We often refer to them as helpers. We know that protein structure is really complex. Let me just fix my volume here. This is actually the structure of a protein, what it looks like in 3D shape, its overall tertiary structure. We're looking at a generalized picture of an enzyme here. So, the flow of energy through life, life is built on chemical reactions. We call it metabolism or energy utilization. All of that energy comes from the sun on our planet, okay? And it's all about how we break that down. So, we're just looking at a quick energy flow here. Sun gives energy to the plants that the giraffe eats. The lion eats the giraffe. The lion gets that energy. So, the chemical reactions of life um, are building molecules, which we call synthesis, and we've talked about dehydration synthesis. So, synthesis, creation of the molecules. And then the breaking down of molecules, which is our digestion, the hydrolysis of these molecules here. Okay. So, how important are enzymes? All chemical reactions in living organisms require enzymes to work. So, the answer to that is they're very important. Um, we are always building molecules, and I put here dehydration synthesis so we don't forget. I had talked earlier about how we lose an OH and we lose an H, and water comes out. However, an enzyme is required for this to happen. It doesn't just happen because of water. So we have synthesis enzymes, but we also have digestive enzymes that help in the process of hydrolysis, which is breaking down these molecules, right? Digestion is just breaking down molecules into their smaller subunits so we can um, utilize them for what we need to utilize them for. So enzymes speed up these reactions. They're what we call a catalyst. A catalyst is something that speeds up a reaction. Enzymes are catalysts. Almost all enzymes are proteins, which is why we're talking about this right after proteins, because the structure of these enzymes is really important to understanding how they work. Okay, so we're just taking a look at some examples. We have our synthesis enzyme, okay, and our digestive enzyme, and it's just putting them together with dehydration synthesis reaction. In a dehydration synthesis reaction, we also refer to as condensation, and then we've put these two subunits together to make this larger molecule. And in our digestive or our hydrolysis that occurs, right, we're breaking them down into the smaller subunits. So each enzyme is a specific helper to a specific reaction. Each enzyme needs to be the right shape for the job. So we're going to learn the different parts of the enzyme, but each enzyme is what we call specific. An enzyme is specific, meaning it only has a shape, a correct shape to work with one particular type of what we call substrate, and we're going to talk about that in a minute. But protein shape is really important to its function. This is one of the reasons why. Okay. Enzymes are named for the reactions that they help. So sucrase breaks down sucrose, which is fructose and glucose together, and table sugar. Proteases break down proteins. Lipases break down lipids. So I want you to recognize ASE. Enzymes almost always end in ASE. ASE, okay? So lipase breaks down lipids. And then we have DNA polymerase, which builds DNA. Okay. So enzymes are not used up in a reaction, okay? They're not changed, they're not used up, and they are recyclable, which is a very important concept. They're only used temporarily. We can reuse them again for the same reaction with other molecules. So very little enzyme is needed to help in reactions. So we're taking a look here. This is an enzyme, this is our protein, and what we're taking a look here is called an active site. What we see here is a substrate. Okay, a substrate is whatever that enzyme is going to react with. So if you take a look at the way this enzyme's active site has a shape that fits one particular substrate, which is why enzymes are very specific. So they join together. So we have the enzyme plus the substrate. They join together for a moment for the reaction to occur. That's called the enzyme substrate complex. And then they bring it through the reaction, so they release the product, and then there the enzyme is again completely unchanged and able to be reused. Oh, lizards. All right. I usually get ahead of myself in these things. So the shape matters. This is a lock and key model, okay? The shape of the protein allows the enzyme and substrate to fit into it, just like I was talking about before. Okay. The specific enzyme for a specific reaction. Okay, and this is just looking at a more advanced picture of what I was showing you earlier. Substrate binds to the enzyme. The enzyme, we take a look at water being added in here. The substrate is converted into its product, so it's breaking it off. I want you to notice that this is hydrolysis occurring. We added the H and the OH, and then the enzyme's free to go do the same thing over and over and over again. Okay, and that's just a closer look. So 1, the active site is available. 2, the substrate binds to the enzyme. 3, with the addition of water, the substrate is converted into its products. Okay. So here's some very important vocabulary you need to understand. The enzyme is the helper protein molecule. It's more biologically appropriate to call it a catalyst. But yeah, the substrate, is the molecule that enzymes work on. Okay, so we're taking a look here. A lot of times I like to say, so here's our enzyme, here's the active site, here, along comes the substrate and it fits in. The enzyme kind of changes shape so that nothing else can bind, only one substrate can bind. I always say the enzyme's giving it a little hug, it's happy to see him, right? We'll bring it through the reaction. The products are what the enzyme produced from that reaction. And the active site is the location that substrate molecule fits into on the enzyme. Okay, so there's various factors that affect enzyme action. Obviously, we need to have the correct protein shape. Okay, when a protein is misshapen, it's denatured. So temperature can affect it, pH, acids and bases. The amount of substrate present is very important. Okay, so based on what we just said in the lecture earlier, a couple slides forward, does the amount of enzyme really matter? The answer is no. Okay, so temperature, what happens if temperatures get too hot? The protein changes shape, its bonds break, changes. If the protein changes shape, then it loses its function. pH, acids and bases can affect it by changing the shape of a protein, denaturing it. The amount of substrate is important because that's what the reaction is dependent on. The speed of the reaction is dependent on how much substrate is there. So here we go. All enzymes have an optimal temperature. So as temperature increases, so does the speed of molecular motion. Okay. The more collisions that occur, the faster the reactions will happen. Enzymes don't have eyes and they're not out looking for their substrate. But if molecules are moving around, they randomly bump into each other and fit in just right, and the reaction occurs. The faster that's happening, the faster the reactions can happen. But if we raise the temperature to boiling, we'll denature the protein will unfold and it will lose its shape, which no longer works. Okay? If we lower the temperature, the molecules will move slower. There'll be fewer collisions between enzymes and substrates. So I want you to think just for a moment, why do we cook our food? Why do we want to cook our food? And then what happens when we freeze our food? So when we cook our food, we're denaturing enzymes. And when we freeze our food, we're preventing any collisions. We're stopping molecular motion. Okay? So we're taking a look at human enzymes here, reaction rate and temperature. So we have what's called the optimal temperature right about here. 37 degrees Celsius happens to be body temperature for humans. Okay? So you can see here that the enzyme will not work at a lower temperature. And right about here is where denaturation occurs. pH, okay, we've learned that pH is the amount of hydrogen ions in concentration. Okay, so changes in the pH can change the protein shape. Most of the human enzymes are between 6 and 8. Okay, and it depends on where it is in the body. Pepsin in the stomach is an enzyme that breaks down proteins, and that has a pH of 3, which makes sense because we have stomach acid, and that has a very low pH. Trypsin is in our small intestines, okay, and that has a pH of 8. So remember, below 7 acidic, above 7 basic. The more acidic, the more hydrogen ions they have. Those hydrogen ions will interact with the bonds of the tertiary structure of the protein and unfold the protein. Okay. So we're taking a look at the same thing here. Okay. This particular enzyme does not have any function at a lower, and doesn't have any function at all above 9. So its optimal temperature is 7. This means it's been denatured or it's been denatured. Okay. Stomach pepsin is much lower. Stomach pepsin won't work above 4 because it's denatured. So this is trypsin in the intestines. Okay. Oh, sorry. This is trypsin in the intestines. And the same thing, when we don't see this curve anymore, it means the enzyme has stopped working. And when an enzyme stops working, it usually means that it has been denatured or we're out of substrate. So either one of those. Okay. Enzyme shape is what matters. We're not going to do that. Okay. So thank you. We will work on this. Let me just stop my video here. Or maybe not. I don't know what's going on here. Okay, sorry about that.